Safeguard systems of DNA. Restriction enzymes, also known as restriction endonucleases, are enzymes that recognize specific DNA sequences and cleave the DNA at or near those sequences. They are an essential tool in molecular biology and genetic engineering, allowing scientists to cut DNA molecules at precise locations. Recognition sequence. Each restriction enzyme recognizes a specific short DNA sequence, typically 4 or 8 base pairs long. The sequences are usually palindromic, means they read the same in both directions. Then cutting patterns. These are two types of cuts made by restriction enzymes: blunt ends and sticky ends. Some enzymes cut the DNA strands at the recognition sequence in a way that produce blunt ends, where the DNA is cut straight across both strands. Other enzymes create staggered ends, resulting in sticky ends. Sticky ends have overhangs single-stranded DNA that can form hydrogen bonds with complementary sequences. Once again, some enzymes cut the DNA strands at the recognition sequence in a way that produce blunt ends, where the DNA is cut straight across both strands. Other enzymes create staggered cuts, resulting in sticky ends. Sticky ends have overhangs single-stranded DNA. Overhanging single-stranded DNA that can form hydrogen bonds with complementary sequences, facilitating the joining of DNA fragments. Then bacterial origin restriction enzymes were first discovered in bacteria. They act as a defense mechanism against invading virus bacteriophages. Bacteria have methyltransferase enzyme that modify their own DNA at the recognition sequence to protect it from being cut by the corresponding restriction enzymes. Then types and nomenclature. Restriction enzymes are categorized into different types based on their properties. The most common type is type 2, which is widely used in research and biotechnology. The names of restriction enzymes usually indicate the bacterial species from which they were isolated. For example, EcoR1 is recognized from Escherichia coli strain RY13, while HIND3 is derived from Haemophilus influenzae strain RD. Restriction enzymes are crucial tools in molecular biology and genetic engineering. They allow scientists to create recombinant DNA molecules by cutting DNA at specific sites and joining them with other DNA fragments. This technique is used in cloning genes, constructing plasmids, generating genetically modified organisms, DNA fingerprinting, and many other applications. Isochiasmers and neochiasmers. Some restriction enzymes recognize the same DNA sequence and produce the same cutting pattern. These enzymes are called isochiasmers. Neochiasmers, on the other hand, recognize the DNA sequence but produce different cutting patterns. Then restriction enzymes. A restriction enzyme, restriction endonuclease, RNase, RNase, or restrictionase. Restrictase is an enzyme that cleaves DNA into fragments at or near specific recognition sites within molecules known as restriction sites. The term restriction enzyme originates from the studies of phage lambda, a virus that infects bacteria, and the phenomenon of host-controlled restriction and modification of such bacterial phage or bacteriophage. Restriction endonuclease recognizes a specific, rather short nucleotide sequence on a double-stranded DNA molecule called a restriction site and cleave the DNA at this recognition site or elsewhere depending on the type of enzyme. Stuart Lin and Werber Arber discovered a restriction endonuclease in E. coli in the late 1960s. These enzymes get their name from the fact that they prevent inversion by foreign DNA, such as viral DNA, by cutting it up. Thus, they restrict the host range of the virus. Many restriction enzymes make staggered cuts in the two DNA strands, leaving single-stranded overhangs or sticky ends that can base pair together briefly. EcoR1 is the first restriction enzyme from the Escherichia coli R strain. Restriction enzymes have to do two things: recognize a specific sequence, cut or restrict at or near the sequence. Restriction enzymes differ not only in the specificity and the length of their recognition sequence, but also in the nature of the DNA strands and the ends they generate. Thus, some enzymes such as HPAL generate flush or blunt ends. Others such as EcoR1, HIND2, and PSTL generate staggered ends. For example, EcoR1 cleaves covalent phosphodiester bonds between G and A at staggered positions on each strand. The hydrogen bonds between the 4 base pairs between these cut sites are easily broken to generate 50 protruding ends of the 4 nucleotides in length. A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequences at specific sites, producing DNA fragments with a non-sequence at each end. And types of restriction enzymes. There are 3 major classes of restriction endonuclease. Their grouping is based on the types of sequence recognized, the nature of the cut made in the DNA, and the enzyme structure. Type 1 and 2 restriction endonucleases are not useful for gene cloning because they cleave DNA at site other than the recognition sites and thus cause random cleavage patterns. In contrast, type 2 endonucleases are widely used for mapping and reconstructing DNA in vitro because they recognize specific sites and cleave just at these sites in. So in addition, the type 2 endonuclease and methylase activities are usually separate single subunit enzymes. Type 1 restriction enzyme, type 2 restriction enzyme, and type 3 restriction enzyme. Type 1 restriction enzymes are also called restriction endonucleases. They are made of 2 long strands of DNA joined together. These restriction enzymes recognize certain sequences of DNA and cleave them at a site. The type 1 restriction enzymes were the first restriction enzymes to be identified. These enzymes are categorized by their DNA cleavage sites. Type 1 enzymes cut DNA far away from the recognized sequence in the DNA molecule. They do not cause effective fragmentation of the DNA and hence are of not much importance. The type 1 restriction enzymes have 3 subunits that perform restriction, digestion, recognition, and also modification of the DNA with the help of its cofactor called magnesium ions and ATP, adenosine triphosphate, that fulfill the catalyzing activity of the enzyme. With the type 1 enzyme, the cleavage site can be up to 1000 base pair away from the recognition site. Then type 2 restriction enzyme. Type 2 restriction enzymes are made up of 4 strands of DNA, 2 on each side of the DNA. Type 2 restriction enzymes are made up of 4 strands of DNA, 2 on each side of DNA. They usually recognize 2 bases on one strand of DNA and cleave the DNA. The type 2 restriction enzymes are vastly different from type 1. For type 2 restriction enzymes, recognition of the sequence and the restriction digestion, the DNA cleavage occurs at the same place. The sites are not different from each other. Moreover, for the cofactors, the type 2 restriction enzymes usually only use magnesium ions for completing the restriction process in DNA molecules. The type 2 type is the most common restriction enzyme available and it is used the most for carrying out restriction. Another major characteristic of the type 2 enzyme is that these enzymes either cut through the middle of the DNA strand, cause blunt ends at both sides, or create cleaves at staggered positions, leaving sticky ends. The type 2 restriction enzymes also have more than just one subunit, and the subunits perform several functions. These are a family of unrelated proteins. They are named after the bacterial species from which they are isolated. For example, EcoR1 is isolated from bacterial species E. coli. For example, the type 2 enzyme is called EcoR1, isolated from E. coli, cuts DNA only at the hexanucleotide. Digestion of DNA with a type 2 enzyme therefore gives a reproducible set of fragments whose sequences are predictable if the sequence of the target DNA molecule is not. The restriction enzymes generate 2 different types of cuts. Blunt ends are produced when they cut the DNA at the center of the recognition sequence, and sticky ends produce an overhang. So type 2 enzymes are distinguished because the recognition site and cleavage site are the same. The sites range in length from 5 to 8 base pairs. The sites are typically inversely palindromic, that is, reading the same forward and backward and complementary strands. Restriction enzymes can cut the DNA in 2 different ways, one demonstrated. Then first and the most common is staggered cut, which leaves single-stranded overhangs or sticky ends. The overhang can be 3' or 5' overhang. The second way is blunt double-stranded cut, which does not leave one overhang. An additional level of specificity determines whether the enzyme will cut DNA containing a methylated base. The degree of specificity in the site also varies. So most enzymes are very specific, whereas some will allow multiple bases at 1 or 2 positions within the same type. 2 restriction endonuclease recognize and cleave DNA within particular sequence of 4 to 8 nucleotides which have a 2-fold axis of rotational symmetry. Such sequences are often referred to as palindromes because of their similarity to words that read the same backward and forward. For example, restriction and modification enzymes of a CoRI, axis of symmetry. Then type 3 restriction enzymes. Type 3 restriction enzymes, these are multifunctional proteins with two subunits, RES and MODE. Before that, we learned about this type 2. Then the DNA sequence specific for the system is characterized by mode subunit. Type 3 restriction enzymes are DNA cutting enzymes that recognize patterns of DNA that are not necessarily based on DNA sequences. Type 3 restriction enzymes are multifunctional proteins. This type of restriction enzymes cuts the DNA away from the recognition sequence. They have two subunits that carry the function of DNA methylation or modification and restriction digestion. These enzymes use the ADOMET cofactors generally for carrying out the restriction process. Then type 3 enzymes cleave at sites a short distance from recognition site, require ATP but do not hydrolyze it. Type 3 enzymes have closer cleavage sites, usually 20 to 30 base pair away. Major kinds of damage to DNA and causes. Damage to DNA consists of any changes introducing a deviation from the usual double helical structure. These are three major classes of DNA damage: single base changes, structural distortion, and DNA backbone damage. DNA can be damaged in many different ways, and this damage, or if left unrepaired, can lead to mutations, changes in the base sequence of DNA. This distinction is worth emphasizing at the outset. The DNA damage is not the same as mutation, although it can lead to mutation. DNA damage is simply a chemical alteration to DNA. A mutation is a change in a base pair. For example, the change from a GC pair to an ethyl GC pair is a DNA damage. The change from GC pair to any other natural base pair is a mutation. If a particular kind of DNA damage is likely to lead to a mutation, we call it genotoxic. Then single base changes. A single base change or conversion affects the DNA sequence but has only a minor effect on overall structure. For example, replacement of the amino group of cytosine with oxygen converts cytosine to uracil, a base that should only be present in an RNA chain. This type of conversion process is called deamination. Deamination is the most frequent and important kind of hydrolytic damage and can occur simultaneously from the action of water or can be induced by chemical mutagen. When the UG base pair replaces a CG base pair, this causes only a minor structural distortion in the DNA double helix. This type of damage is not likely to completely block the process of replication or transcription, but only lead to the production of a mutant DNA or protein product. And vertebrate DNA frequently contains 5-methyl cytosine in place of cytosine. Methylated cytosines are hotspots for spontaneous mutations in vertebrate DNA because deamination of 5-methyl cytosine generates thymine. This results in the change of a GC base pair into an AT and damaged DNA is replicated. DNA is also subject to damage by alkylation, oxidation, and radiation. Alkylating agents such as nitrosamines lead to the formation of O6-methyl guanine. This modified base often mispairs with thymine, resulting in the change of a GC base pair into an AT base pair when the damaged DNA is replicated. So, potent oxidizing agents are generated by ionizing radiation and by chemical agents that generate free radicals. This reactive oxygen species, for example O2H2, O2OH, can generate 8-oxoguanine, a damaged guanine base containing an extra oxygen atom. Oxo-G is highly mutagenic because it can form a Hoogsteen base pair with adenine. This gives rise to a GC-TA conversion, which is transversion, which is one of the most common mutations found in human cancers. Then structural distortion. Ultraviolet light has a detrimental effect on cells due to the selective absorption of the UV rays. Radiation with a wavelength of about 260 nm is strongly absorbed by the bases. The most frequent UV-induced lesions of DNA are the induction of pyrimidine dimers between two neighboring thymine bases by UV radiation. These are also termed cyclobutane pyrimidine dimers because a cyclobutane ring is generated by links between carbon atoms 5 and 6 of adjacent thymines. Because covalent bonds from between thymines on the same strand, this disrupts the complementary base pairs that form the double helix. Thymine dimers thus distort the structure of the duplex DNA. Then structural distortion may impede transcription and replication by blocking the movement of polymerase. Consequently, the reduction of pyrimidine dimers is a more severe defect than a single base change. UV radiation can also induce dimers between cytosine and thymine called pyrimidine, and other bulky adducts can be induced by chemical mutagenesis. By exposure to large polycyclic hydrocarbons or alkylating agents. The DNA damage is also caused by intercalating agents and by base analogs. Intercalating agents such as ethidium bromide contain several polycyclic rings. Thus, flat rings insert between the DNA bases, binding and stacking within the DNA bases, just as the base bind or stack with each other in the double helix. Due to the resulting distortion of the double helix, intercalating agents can cause insertion or deletion of one or more base pairs during DNA replication. Base analogs are compounds such as 5-bromouracil, an analog of thymine that substitute for normal bases. They are similar enough to be normal bases to be taken up by cells, converted into deoxyribonucleotides and incorporated into DNA during replication. However, because of their structural differences, analog base pair inappropriate. For example, 5-bromouracil can be paired with guanine. Then DNA backbone damage. Backbone damage include the formation of abasic sites, loss of the nitrogenous base from a nucleotide, and double-stranded DNA breaks. Abasic sites are generated spontaneously by the formation of unstable base adducts. For example, in purine nucleotides, the sugar-purine bonds are relatively labile. So, hydrolysis of N-glycosyl lineage in the purine base by the action of water leaves the hydroxyl in its place in the depurinated DNA. Double-strand breaks can be induced by ionizing radiation, X-rays, radioactive materials, and a wide range of chemical compounds. Ionizing radiation can attack double-strand breaks can be induced by ionizing radiation, X-rays, radioactive materials, and a wide range of chemical compounds. Ionizing radiation can attack or ionize deoxyribose sugar in the DNA backbone directly or indirectly by generating reactive oxygen species. Double-stranded breaks are the most severe type of DNA damage since they disrupt both the DNA strands. Damage caused by alkylation of bases. Alkylation is the reaction of electrophilic chemical compounds or alkylating agents with the nucleophilic centers in organic macromolecules. Alkylating agents include a large variety of chemicals, some of which are potent mutagens or carcinogens. Alkylating agents include a large number of molecules which can effectively and efficiently react with the DNA, causing structural modification of the bases and phosphate groups in DNA. The most common agents are alkyl sulfates, alkyl sulfonates, alkyl halides, dialkyl nitrosamine, alkyl nitrosols, and acyl nitrosamides, mustards, diazo compounds, lactones, epoxides, etc. These agents can be divided into two major groups, monofunctional and bifunctional. The monofunctional agents have only one reactive group which is involved in covalent interaction with the single center on DNA. The bifunctional agents have two reactive groups and have an ability to react with the two centers on DNA. If the two centers are on the opposite strands of the DNA, the reaction of bifunctional agents such as sulfur or nitrogen mustards can produce an ultra-strange cross-link. Many of the alkylating agents have a mutagenic effect and are suspected carcinogens. The reaction specificity of these compounds is different in different bases. Diazoalkyls react readily with guanosine and thymidine but only under extreme conditions with adenosine and cytosine. In contrast, alkyl sulfates, alkyl sulfonates, mustards, epoxides, and nitroso compounds alkylate bases in the following order. The rate of ethylation is much lower than that of methylation for all classes of alkylating agents except alkyl iodides, which readily alkylate G, A, and C. Many environmental carcinogens or cancer-causing agents are electrophiles that act by attacking DNA and alkylating it, as we have just seen. This can lead to mutations. If the mutations occur in genes that control or otherwise influence cell division, they can cause a cell to lose control over its replication and therefore change into a cancer cell.